My idea here is that you can use a picture to show the difference between these two things. Mono means one, poly means many, and saccharide means sugar. So most of you recognize that a monosaccharide has 1 unit of sugar. Someone asked me while I was walking around, could I say 1 atom? And I want to make sure you all understand you couldn't. A unit of sugar is a whole bunch of atoms put together. Usually sugars look like a hexagon or sometimes like a pentagon if we were to just draw their basic shape. Now that's me ignoring that that's a bunch of carbons and hydrogens and oxygens all attached to each other. We're just doing the simplest representation possible. So a polysaccharide could be 2 or more of these put together. So I'm just drawing 2 for space sake, but polysaccharide means many. And so we would probably want to say, there's 3 or 4 or 5 or 100 of these. But as long as you get the idea, that's how you can tell the difference between them. So if on Friday, when we do our first major quiz, which I like to refer to as a quest because it's not a little baby quiz like we do at the beginning, it's not a test like we do at the end, when we do that quest, if I were to show you a picture, you could tell what type it is just by looking for this basic structure. Now I noticed a number of people were stuck on glycogen and starch until I said they come from different organisms, and then quite a few of you remembered that glycogen comes from an animal and starch from a plant. So you very easily could have drawn a person and a flower to represent the difference. Glycogen is the storage form for a carbohydrate in your muscles. Starch is a storage form for plants. Now, dehydration, synthesis, and hydrolysis, I think there are a number of ways that you could use based on what I saw people doing. Some people focused on whether I was gaining or losing water. So you could say, in dehydration synthesis, I lose a water. In hydrolysis, I add a water. That would be a perfectly acceptable way to show the difference between them. You could also focus on what happens. Now, I saw a few people sketch out the little shapes that we were cutting out last week. And so if you showed, for example, 2 of these shapes going together, that's synthesis. Those 2 pieces coming apart would be hydrolysis. So you could represent what's happening with water, am I gaining or losing it, and what's happening with the molecule. And they both would be acceptable ways to talk about what's different. Now, saturated and unsaturated fat have a couple of things that you could use to distinguish between them. A saturated fat does not have double bonds between carbons. So you could very simply put something like this. One of them has single bonds between carbons, the other has double bonds. You could also put a picture of what the fat would look like. So I showed you last week when we were talking about glycerol and fatty acids. If this piece here is glycerol, it's the backbone, there would always be 3 fatty acids attached to it. If they look like a straight line, those are usually saturated. And when it becomes unsaturated, it'll usually cause a bend or a kink in the shape. So that would be something about the actual molecule that you could look for. You could additionally say that saturated fats are usually the harder kind, more solid kind. Unsaturated fats are typically softer. Sometimes they can even be in liquid format. Now the next one here is a little bit harder to represent with a picture if you want to talk about a protein specifically. The idea here is that these are both changes that happen to a protein structure. Coagulation is permanent, and denaturation is temporary. And so what I was going to draw here was some representations of things that are permanent and things that are temporary. So for example, if I draw a Sharpie versus a pencil, that is the idea of permanent versus temporary. I can undo denaturation. Now, as I was walking around, I saw that someone for denaturation showed water turning to ice and ice turning to water. It's not the exact same thing, but the idea of reversibility is really what we're getting at. In denaturation, you can reverse it. My analogy, of course, for denaturation was a cassette tape that I could wind back up after the tape had been pulled out of it. Whereas my example for coagulation was cooking an egg, and I saw quite a few people drawing eggs as I walked around. Now the last thing on the list, anabolism and catabolism, are the two types of metabolism that an enzyme could help achieve. Now the difference between them is, am I making something or am I breaking something? So anabolism would look something like this. I take two pieces, I put them together. Catabolism would be the same thing, but the other way around. Now on Friday, I gave everyone coloring as your homework, and this is what you had to recognize, was that one of the enzymes was showing two small substrates being put together to form one product. That's anabolism. The other enzyme had one large substrate that got broken up into two smaller products. That's catabolism. Now, would anybody like to ask any questions about anything that's on here? So the last thing that we're going to talk about are factors that affect enzyme activity. Now, I want to remind you all, before we write anything else, that enzymes are proteins. So anything that could affect a protein could also affect an enzyme because that's what they're made out of. So the first factor is temperature. Does anyone have any idea whether temperature makes chemical reactions go faster or slower? Yeah, it generally makes them go faster, but we're going to add a little caveat at the end of that. Temperature increases activity to a maximum. Now, I can't give you a specific number because not every enzyme functions the same way. But I will mention, for example, that your body should be in the neighborhood of 37 degrees Celsius. And most of the enzymes that we're going to discuss are enzymes that you have personally in your human body. And so most enzymes that we're going to discuss function best at or around body temperature. So temperature will increase enzyme activity to a maximum. Beyond this, what will happen to an enzyme? If it gets too hot, what are two things that we just did in the daily review that might happen to my enzyme? One of them is permanent, one of them is not. Well, somebody knows these words. You all just wrote them down, all 22 of you. All right, why don't you tell me one of the words? So, beyond this, coagulation and or denaturation could occur. So temperature will help out an enzyme. Generally, things being warmer means they work faster, and that's because particles move more quickly when they're warmer. But beyond this, coagulation and or denaturation depends how hot it is. Remember that coagulation is permanent and denaturation is temporary. So denaturation would happen first, coagulation would happen second. It would happen as it got even hotter. So just so that you can see what this might look like on a graph, which is a really good way for me to ask you about enzyme activity, the first little sketch I have on the left is what it would look like if I tried to graph an enzyme's activity compared to temperature. If you look at this first little graph, I have temperature in degrees Celsius along the bottom, and enzyme activity along the side. As you can see, the line goes up very steadily and then really sharply drops off. And that's because we got past that maximum point. Look at how the maximum there was very close, or exactly 37 degrees Celsius, and then the enzyme really quickly stopped functioning. Would anyone like to ask anything about temperature here? So then the next thing is concentration, and I'm actually going to talk about two different things. Concentration of the enzyme and concentration of the substrate. Now when we talk about concentration of the enzyme, the point that I want you to remember is that enzymes can be reused. They don't actually participate in the chemical reaction. They help a reaction happen faster, but they're not altered, they're not broken apart, we're not putting them together. Because enzymes are reused, changing the concentration of the enzyme will have little to no effect. As long as you have enough enzyme, adding more isn't really going to make a humongous difference. So changing the concentration of the enzyme isn't usually the thing that has the effect. Changing the concentration of the substrate will have a huge effect. The substrate is the thing that's actually reacting. If I have more molecules that can react, well, that means I can have more reaction. So if I increase the concentration of the substrate, I will have a faster reaction. The enzyme will have more substrate molecules to work with, and there won't be sort of a pause or a delay while the enzyme looks around for another molecule. There will be lots of them. So increasing the concentration of the substrate will increase activity. We will have a faster reaction. Now the third thing on the list is pH. And pH is very similar to temperature, in that there's a maximum, there's an optimum pH that each enzyme works at. So all enzymes have an optimum pH. More or less, so if I go above the optimum pH or below the optimum pH, decreases activity. So I put a little illustration at the bottom, this one right here, that shows that not every enzyme has the same optimum pH. Some enzymes, like the one on the left, have a spike really close to the edge. That means that when the pH is really low is when they work their best. So things that work in your stomach, and we'll worry about the names of these when we get to the unit on your digestive system, but the enzymes that are in your stomach will generally have their optimum pH at a really low number. Whereas other enzymes, like say this one over here, that has a much higher pH as its optimum, this would be more like the enzymes that work in your intestines. They need a much higher pH to function. Notice how I didn't put any actual numbers here, because that's not what I want you to recognize. I want you to recognize the pattern. Some enzymes function best at a low pH. Some enzymes function best at a high pH. Now the last thing on the list are inhibitors, cofactors, and coenzymes. These are all substances. They could be individual elements, possibly. They don't have to be. They could be bigger molecules. But this is another type of molecule that we're going to add to the mix. Now, obviously, inhibitors will decrease activity. Cofactors and coenzymes increase. And you can sort of tell that just by the way the word is written. To inhibit something means to make it stop, not happen. And whenever you have the word co at the beginning of a word, like cooperate, it means that they're going to work together. So cofactors and coenzymes increase enzyme activity. Now there's a couple of pictures down here of where these things could attach themselves. It's possible, like in this picture, for the substance that we're talking about to attach to the enzyme somewhere other than the active site. Now the types of molecules that do that, if it was an inhibitor and there was an element or a molecule that attached somewhere other than the active site, we would call this a non-competitive inhibitor. If it was helping, we would call it a cofactor. So a molecule could attach to an enzyme, not at the active site. If it helps the enzyme, we'd call it a cofactor. If it doesn't help the enzyme, if it slows its activity, we would call it a non-competitive inhibitor. The other thing that's possible is that this molecule attaches to the active site. So not somewhere else, but actually to the spot where molecules are supposed to be fitting into this enzyme. This is where we would have a competitive inhibitor. A competitive inhibitor would block the active site. It would fill it up so that another molecule couldn't bind there. We could also have what we call a coenzyme, and we'll actually talk about a number of coenzymes as we go through this unit. A coenzyme would bind to the active site, but instead of blocking it, it would complete it. It would make it the correct shape so that another molecule could attach. So of the substances that could bind to an enzyme and change its activity, we have cofactors and coenzymes that increase enzyme activity, inhibitors that decrease, and what they are depends on whether or not they're attaching to the active site.